These are the goals for the DNA Replication Lab unit. After watching this video and using the other online resources, you should be able to address these goals. Follow along in your manual and take notes as we go through the process. This figure shows diagrams of eukaryotic DNA. We can tell that this is eukaryotic DNA because it is linear, not circular as in prokaryotes. In addition, there are multiple origins of replication. This diagram shows a simplified view of DNA replication with most of the important molecules shown. Now, while our lab simulation would not utilize all of these, you should know these molecules and their functions for lecture. In lab, we would have used a DNA model kit to simulate DNA replication. In your manual, you can see each piece that would have represented the 4 different nucleotides. You can also see the orientation of each piece and how each piece represents the 5' phosphate group, the 3' hydroxyl group, and the nitrogenous base. This slide and activity reinforces the base pairing rules and the 5' to 3' orientation of the 2 DNA strands as they run anti-parallel to one another. Here is a sample DNA molecule. Well, one strand of it anyway. Pause the video here and write this molecule on a piece of paper. Then write down the complementary strand. What would the second strand look like? Right, so here is our complete DNA molecule. Now let's practice the steps of DNA replication. Here we can see the 2 strands of DNA have been separated. What enzyme was responsible for doing this? Right, helicase. We can show helicase here, separating the hydrogen bonds between the nitrogenous bases. Now our 2 strands would go right back together again if single-stranded binding proteins didn't hold the 2 strands apart. Now take a look at our 2 strands. We can see that they run anti-parallel to one another, right? But DNA replication can only occur in one direction. Which direction is that? Yes, DNA replication can only occur in the 5' to 3' direction. So that means DNA replication has to occur in this direction on this strand, and in this direction on this strand. Given this information, we can determine which strand is the leading strand and which strand is the lagging strand. Can you tell which is which? That's right. DNA polymerase III can only add nucleotides to an existing 3' hydroxyl group. Because of this, we need an RNA primer for DNA polymerase III to add DNA nucleotides to. What enzyme is going to make the RNA primer? Right, primase. The primer is shown here in red. And here are the prime ends. We can see that our primer is oriented anti-parallel to the parent strand as well. Once primase has added a small RNA primer, DNA polymerase III can begin adding DNA nucleotides to the 3' end and synthesize the leading strand. On the leading strand, DNA polymerase III will follow helicase as it separates the 2 strands. The new DNA strand is shown here in green. Do you remember the name of the bonds that form the sugar phosphate backbone of the DNA molecule? That's right, phosphodiester bonds. Now the lagging strand is also synthesized in the 5' to 3' direction. However, since the 2 DNA strands run anti-parallel to one another, the lagging strand is synthesized moving away from the replication fork and helicase. This is why the lagging strand is made in pieces, or fragments known as Okazaki fragments. Each fragment needs its own RNA primer before DNA polymerase III can add the DNA nucleotides. This process will occur over and over again as helicase continues to expose more of the parent DNA strands. Once the 2 parent strands have been replicated, the RNA primers must be replaced with DNA nucleotides. DNA polymerase I will do this job. The primer on the leading strand will be replaced, as well as the primers on the lagging strand. Finally, we have to make sure all of our fragments are connected. DNA ligase will form the phosphodiester bonds between the fragments so that we have 2 complete new strands of DNA. This is the end of the DNA Replication Lab simulation. Be sure you have completed all pre-lab and post-lab questions, as well as questions throughout the chapter.